Hi, everyone, and welcome back to the next webinar in our Plantae Presents Global Plant Science Talk series. My name is Katie Rogers, and I'm your host for today's webinar. This webinar series is brought to you today by Plantae, the open online community for plant scientists powered by the American Society of Plant Biologists. I would like to give a special thank you to all of our ASPB members who are attending today. Your ASPB membership dues help support and make these webinars possible. For any of you who have not yet joined ASPB, you can join today and use the discount code PRESENTS10 to receive a 10% discount on registration. ASPB members get early access to these seminars. You can learn more about ASPB and the opportunities we provide at aspb.org. So far, we've held over 35 events in this series. If you haven't already done so, check out our archive to watch all of the previous talks. You can find information about each event on our plantae.org blog and subscribe to our Plantae YouTube channel to get notifications when new recordings are posted. We have 2 more weekly events lined up through April. After that, we'll be switching gears to plan and host the annual plant biology meeting online. Next week, oops, I don't have a slide, but next week we have a special event featuring 3 panelists who have recently published in the Plant Cell Focus issue on the biology of plant genomes. You can find information to sign up for this event on our blog at plantae.org. Hope to see you there. If you need to leave this webinar early, know that a recording will be made available within a few days. Today's talks will each be 13 minutes, followed by a short question and answer section. As you listen to each talk today, please add questions for our panelists in the Zoom question and answer Q&A section. Questions specifically for our panelists should be shared within the Q&A section to make it easier for our moderator to navigate. You'll notice that there's also a chat area open. This section is for you to introduce yourself, say where you're from, and connect with your colleagues. Today, Michael Skelly is with us to moderate. Michael is a postdoc at the University of Edinburgh. Michael's research has shown that the immune activator NPR1 is dynamically regulated by ubiquitination and continues to study both ubiquitin signaling and transcriptional regulation. Michael is an assistant features editor for plant physiology. All right, Michael, I'm going to hand it over to you.
Thanks very much, Katie. It's a great pleasure today to be moderating this session, and we've got 2 excellent speakers who both will touch on, I think, a bit of ubiquitin signaling, which is a personal interest of mine. So our first speaker is Professor Sophia Stone. She's the George S. Campbell Chair of Biology at Dalhousie University in Canada. She has a long-standing interest in the ubiquitin-proteasome system, and today she's going to speak to us about how the ubiquitin-proteasome system is at the crossroads of abiotic and biotic stress tolerance. So welcome, Sophia. I'll hand over to you now.
Thank you, Michael, and thanks to ASPP for inviting me to speak today, and thanks to all of you for listening to my talk. So I'll share my screen now. Okay, so I'm going to talk about briefly the ubiquitin-proteasome system, and then I will go into highlighting 2 ring E3 ligases that play a role in both biotic and abiotic stress responses. So for the ubiquitin-proteasome system, you have 2 main events. So ubiquitination followed by degradation of the modified substrate. And ubiquitination involves 3 enzymes, the E1, E2, and the E3, which covalently attaches ubiquitin to a lysine residue within the substrate. Now, ubiquitin itself has 7 lysine residues, and each can be used to build ubiquitin-ubiquitin linkages to generate a chain. And the modifications collectively are referred to as the ubiquitin code. And each modification is linked to a particular biological process. For example, lysine 48 linked chains are targeted to the proteasome for degradation. Now, this is a dynamic process. The E2, along with the E3, which selects the substrate, writes the code, and then deubiquitinating enzymes can modify the code or erase the code by removing ubiquitin molecules. Now, the main outcome of ubiquitination is actually degradation by the proteasome, and lysine 48 linked chains are the most common type found. And the Picard lab a few years ago, a while ago, showed that you need 4 ubiquitin molecules in a chain for recognition of the substrate by the 26S proteasome, leading to degradation. But recent evidence support what's called the ubiquitin threshold model, where short diubiquitin chains or branched ubiquitin chains are better signals for degradation compared to the canonical lysine 48 linked tetraubiquitin. So a really excellent example for the ubiquitin proteasome system in stress is the abscisic signaling pathway. So just to describe ABA signaling, without stress, the ABA responsive kinases are inhibited by a suite of phosphatases. Upon stress perception, ABA is produced, it binds to its receptor, and it sequestered the phosphatases. So then the kinases can become active, and you have activation of a suite of downstream targets that mediate the response. So layered on top of this regulation is the ubiquitin proteasome system. So you have a number of E3 ligases that target components for degradation, for signal inhibition. Once the stress is perceived, you have E3 ligases that target inhibitory components for activating the signaling pathway. And during the stress response, you have E3 ligases that target activated components to attenuate the signal. So what we study in the lab are a specific subgroup of ring E3s, and these are the ring types. So they're all defined by a ring domain. And while in the Callis lab at UC Davis, we identified 457 ring E3s, and we categorized them into 7 different groups based on variations within the ring domain. A recent publication from the Guzman lab has updated the ring family to include 508 ring E3s. So what we're trying to answer right now is how involved are the ring E3s in stress responses? So what we did was we looked at expression data in microarrays, and we also did literature searches. And we find that 80% of the ring E3s are differentially expressed in response to a stress. And we only look for those who had a twofold change in expression compared to the respective controls. And what I'm showing you here is the DEGs associated with each abiotic and biotic stress. So we then wanted to know the extent of sharing of the ring E3s between biotic and abiotic stresses. So we compared their DEGs, and we found 218 DEGs that are shared between 1 or more biotic and abiotic stresses. So these numbers here just highlight the shared DEGs between each of these stresses. So if we look at the shared DEGs, what we see is that you have similarity in regulation. So if we focus on an example of ozone, bacteria, and fungal infection, and we looked at the DEGs that are shared across all 3 individual stresses, we see that over 50% of the genes are upregulated. So the question that we ask next is, how is it that a particular ring gene can function in all of these individual stresses? and it comes down to what are its substrate. So to try to find substrates, we are building an interaction network for the entire ring E3 family, and we have so far found over 15,000 interactions for 210 ring E3s with 842 proteins. And Go analysis places most of those 842 interacting protein within the category of response to stress. And what I'm showing you here is an example for the SYNAT E3 family, and you can see that each E3 interacts with multiple substrates. And in fact, within the entire network, what we see is that most E3s interact with 2 or more proteins. So this allows each ring E3 to regulate different substrates, different biological processes based on its substrate. So what I'm going to do next is just highlight those 2 ring E3s that I've talked about. And the first is KEG. And this E3 was identified in the Callis lab at UC Davis some time ago. And the first substrate identified for KEG was the ABA responsive transcription factor ABI5. Now, the KEG mutant, it overexpresses, or not overexpresses, but accumulates ABI5. But unlike other plants that also overexpress ABI5, the KEG mutants undergo growth arrest similar to ABA-treated wild-type seedlings. And this growth arrest is partially explained by the accumulation of phosphorylated forms of ABI5 in the KEG mutant. And we have since shown that a member of the SNRK3 kinase family called CIPK26 is also a substrate for KEG. So KEG inhibits ABA signaling by targeting a kinase, as well as a transcription factor for degradation. And further work in the Callis lab has shown that KEG also regulates other ABA responsive transcription factors. So I'm going to switch now to talking about pathogen defense. And Roger Ines's lab at Indiana University was the first to show a role for KEG in pathogen defense. And what he showed is that a mutation in the HERC repeats here, the KEG4 allele, basically suppresses the resistance to powdery mildew that's conferred by the EDR1 mutation. And a recent publication from Deng Zhong Tang's lab shows that the KEG4 mutant is more susceptible to powdery mildew compared to EDR1 or Columbia. Now, the thought is that the KEG4 mutation here does not inhibit the E3 function. What it does is it seems to stabilize the E3 or activate the E3. So KEG was recently shown to ubiquitinate 2 MAP kinases, MKK4 and 5. And both of these kinases are involved in resistance to bacterial and fungal pathogens. And when you look at the accumulation of MKK4 or 5 in the KEG4 background, you can see that you have less of the kinase compared to wild type. And this can be explained by the fact that the KEG4 mutation activates the E3. And the lower levels of the kinase would explain the susceptibility of the KEG4 mutant to powdery mildew. So basically, what we're seeing is that KEG inhibits both biotic and abiotic stresses by targeting signaling components for degradation. So to mount a response, you would need to inhibit KEG activity, right? So here I'm showing results again from Roger Ines's lab. And what he's shown is that in powdery mildew infected cells, the level of KEG decreases, right? And this is a protein degradation assay. So we use cyclohexamide to assess protein turnover in the absence of synthesis. And what we see is that over time, the level of KEGG decreases in the presence of ABA. But interestingly, the KEGG mutant, which has a mutated ring domain, so it's an inactive E3, is quite stable in the presence of ABA. So this tells us that auto-ubiquitination is used to suppress KEGG activity in response to biotic and abiotic stress. So the question is, what is the underlying mechanism that switches KEGG from substrate ubiquitination to auto-ubiquitination? And the obvious answer is phosphorylation, because phosphorylation has been shown to activate E3s, promote auto-ubiquitination, and regulate substrate specificity. And we have shown previously that if you inhibit kinase activity, you can stabilize KEGG in the presence of ABA. So this tells us that you would need to increase phosphorylation in the presence of biotic or abiotic stress so that you can promote auto-ubiquitination and decrease KEGG levels. So what we did to try to identify KEGG phosphorylation is to isolate KEGG from seedlings that are expressing a activated CIPK26. And we took this approach because we know that the activated CIPK26 is stable. It does phosphorylate KEGG, and it does promote KEGG degradation. And so using this approach, we were able to identify one phosphorylation site here within a hERG repeat that was not found in the control. So this is a bit promising. So we're now following up on that phosphorylation site to see if it's actually involved in regulating KEGG activity in the presence of biotic or abiotic stresses. All right, so I'll stop talking about KEGG for now, and I'll switch to the next E3 ligase, which is XBAT35.2. And this is an interesting E3 because it regulates both pathogen resistance and abiotic stress tolerance by modulating the activity of a single substrate. Okay, so XBAT35 mRNA undergoes exon skipping. This exon here, exon 8, is omitted from the XBAT35.2 transcript. And this results in a loss of nuclear localization signal from the XBAT35.2 protein. So XBAT35.1 localizes to the nucleus, whereas XBAT35.2 is Golgi localized. And what we've seen here is co-localization between XBAT35.2 and ACD11, which is accelerated cell death 11. And bimolecular fluorescence complementation shows interaction between the two proteins in tobacco cells. Okay, so the accelerated cell death mutant, it undergoes programmed cell death, and it constitutively expresses a number of pathogen-related defense genes. So if XBAT35.2 is actually targeting ACD11 for degradation, if you overexpress the E3, then ACD11 levels should be suppressed or inhibited. And so this should hopefully lead to cell death or promote pathogen defense. And what we see is that if we transiently overexpress XBAT35.2 in tobacco cells, you can induce cell death. And if you overexpress XBAT35.2 in Arabidopsis plants, these plants are less susceptible to bacterial infection compared to wild type. And we also see increased expression of the pathogen-related gene PR1 in the XBAT35.2 overexpressor. So it does seem that XBAT35.2 is targeting ACD11 for degradation. Now, correlating with these findings is the fact that after pathogen infection, we see an increase in XBAT 35.2 levels, and we see a decrease in ACD11 levels post-infection. So this is at the 0 time point, and this is 12 hours after infection. And we also see change in ubiquitin abundance that corroborate these results. So in mock treated, you can see that you have ubiquitination of XBAT 35.2. And after infection, you can see that levels of ubiquitination decreases. And you have the opposite for ACD11. You have an increase in the ubiquitination of ACD11. Now, to detect ubiquitinated proteins, we use a ubiquitin antibody. And so if you remember that multiple ubiquitin molecules is attached to each substrate. So you cannot resolve them on the gel. So for ubiquitination, what you see is this, basically this smear, this black smear. Okay, so this is our model so far. With pathogen infection, you stabilize the E3 and you get degradation of ACD11 to promote defense, right? So what about abiotic stress? What we see is that XBAT 35.2 levels increase after treatment with, for example, increasing concentrations of salt. So the level of XBAT 35.2 increases gradually along with that increasing concentration of salt. Now, ACD11 levels, it's a little bit more dynamic, right? At early time points, you see this increase in ACD11 levels with salt, but over time, you see a decrease in protein levels. And the decrease is more drastic with higher concentrations of salt. So we then wanted to find out if the increase in ACD11, the increase in E3 that you see here, does it promote the degradation of ACD11 with high salt? And the answer is basically yes, it does. So we assessed protein levels in the presence and absence of the E3, right? And so this is our untreated control. You can see that with and without the E3, ACD11 levels decreases gradually. And this tells us that XBAT 35.2 is not involved in the degradation of ACD levels under these conditions. With high concentrations of ABA or salt, we see a more rapid decrease in ACD11 compared to the control. So ABA and salt is inducing turnover, but this turnover is slowed in the absence of XBAT 35 because you see more protein remaining over time. So this tells us that XBAT 35 is required for the ABA and salt induced degradation. So we next wanted to know if that initial increase in ACD11 is required, or is it relevant to abiotic stress? Does it actually play a role? So what we did was we compared the stress tolerance of transgenic plants overexpressing ACD11 to wild type. And on high salt concentrations, we can see that wild type has a survival rate of about 40%. You have a lot of little white seedlings. Whereas for the overexpressor, you have a high survival rate of about 70%. And the ACD11 overexpressors are also more tolerant to drought stress compared to wild type. The survival rate after rewatering for ACD11 overexpressor is about 90%, compared to only 20% for wild type. So this is the model for under abiotic stress. When stress is perceived, ACD11 levels go up to promote the response, and that gradual increase in XBAT 35.2 promotes its degradation. So putting it all together, we see that under non-stressed conditions, XBAT 35.2 is unstable, and other E3s would regulate the levels of ACD11 to maintain the appropriate amount. Under biotic or abiotic stress, you have stabilization of the E3. For biotic stress, it targets AC11 for degradation to promote the response. So the E3 in this case is a positive regulator. For abiotic stress, it targets AC11 for degradation to attenuate the response. So the E3 is a negative regulator. So overall, what we're finding is that a significant number of the ring E3s are involved in both biotic and abiotic stress responses, and its role as a positive or negative regulator will depend on the substrates that they're targeting. So I'll stop there, and I'd like to thank my lab members, past and present, who has participated in the XPAT 35 and KEG project, as well as collaborators that has helped us with our work. So I will leave it there, and I'll turn it back over to the moderator, Michael.
Thanks very much, Sophia. Fantastic talk. We probably have time for a couple of questions. So first one is from Deru Liu, and they're asking if the XBAT 35 mutant is more susceptible to pathogens.
Yes. Yes, it is. We did show that. I didn't talk about it because of timing, but it is more susceptible to the pathogen compared to wild type and the overexpressor.
Okay, I had a question also related to XBAT 35. So you showed that upon DC 3000 infection, the levels of XBAT 35.2 go up, but levels of ACD11 go down. Yeah. Do you know if Pseudomonas has any proteins that interfere with this process? It's well known that bacterial pathogens can interfere with host ubiquitin signaling.
No, we don't know that. We've not gone that far into the analysis. So all we do know is that the E3 is stable, and then that stabilized E3 can then target AC11 for degradation, but we don't know the interaction with bacterial effectors or anything else.
Okay. We've just had another question come in. Somebody is asking if you know what controls alternative splicing of XBAT 35.
No, no, we don't. Both transcripts are expressed equally in every tissue type, and under different stresses, it's been shown that they're expressed equally. So under control and stress treatments, the level of both transcript increases. So we think that they're both constitutively produced under all conditions, but it's regulated at the protein levels, right? So whether or not the produced protein is stable, and do they become stable under a particular condition? And it's also regulated by localization, because one goes to the nucleus, one goes to the Golgi, and then they might be unstable and then become stabilized depending on the external stimuli. So the regulation is, we don't think is at the transcript level, because they're both constitutively produced.
Okay, that's interesting. Thank you. We have time for two more quick questions. Catherine Dunning's asking, does XBAT 35 work with different E2s under different stress response pathways?
We're not sure what E2 it works with, but we've looked at interaction, and we've seen interaction with the usual UBC8, which everybody uses for ubiquitination assays. But we've also seen interaction with UBC34 family members, but we don't know if those are bona fide or relevant, useful interactions or not.
Okay, thanks. And we'll finish with this one question from Scott Hayes. He's asking, E3 ligases dictate the specificity of ubiquitination. How can some E3s target so many different proteins? Is there a common recognition motif? So this is maybe in reference to KEG, I'm imagining.
Right. So we're not sure what the recognition motifs are, but they won't be common. They'll most likely be different. But the main point of regulation is with post-translation modification of the E3 and the substrate. So depending on how the modifications present on the E3 and the substrate, that determine if they engage one another or not, right? So you have an external stimulus come in. It will either target the substrate and or the E3. Then you have changes in PTM patterns, and then those changes will then allow the interaction between the E3 and substrate, and then you'll get ubiquitination, right? So for the E3, that phosphorylation can either block or promote interaction with the substrate, or it can either promote auto-ubiquitination, for example, so you get rid of the E3, or it can help to stabilize it so the E3 is now present and able to interact with the substrate.
Great, thank you very much. There is another couple of questions, but I'm sorry we don't have time to get through them, but maybe Sophia can see these in the Q&A box, and maybe if she has time she can type an answer to these. But we should probably move on now to our second speaker. And our second speaker is Dr. Sjon Hartman, who is now a Rubicon Fellow at the University of Birmingham. Sjon was previously at Utrecht University, and he recently moved internationally during the pandemic, which is to be applauded. Sjon has very recently just been awarded the Hugo de Vries Prize, which is for the best plant science PhD thesis in the Netherlands. So this is a very prestigious award. So we're lucky to have a celebrity talking to us today. And I'll hand over to Sjon now. He's going to tell us a bit about how ethylene inhibits proteolysis of oxygen sensing proteins to improve hypoxia stress tolerance. Over to you now, Sjon.
Yes, thank you, Michael, for the introduction. So good morning or afternoon or evening, everyone, from wherever you're watching. It's an honor that you are here to watch this this seminar. I want to thank the organizers for giving me the opportunity to present our work alongside Sophia. So for today's theme, to fit in a bit, I've tried to focus a little bit more on the role of proteostasis. And specifically, I would like to tell the story of how the flooding signal and plant hormone ethylene limits the degradation of oxygen sensing transcription factors, so these group 7 ethylene response factors, and in turn mediates or acclimates plants to survive low oxygen stress or hypoxia stress. So I've already, since my bachelor, I've been interested in the signaling dynamics during flooding, and I believe this is very important because floods lead to a vast majority or to a very large proportion of crop losses annually. And I think if we want to really be able to develop flood tolerant crop varieties in the future, we need to understand basically 3 things. And the first one is, why do plants die underwater? What kind of pathways or mechanisms do plants have to survive underwater, so to prevent drowning basically? And how are these pathways and mechanisms or processes regulated? So the first one is actually quite easy, because just like us, plants actually need oxygen, and underwater oxygen supply will ultimately run out because underwater gas diffusion is reduced around 10,000 times. It's 10,000 times slower, and this prevents oxygen going into the cells, ultimately leading to oxygen deprivation or so-called hypoxia. Therefore, the capacity to survive hypoxia or the oxygen deprivation is a very strong determinant of flooding tolerance in plants. Now, kind of paradoxically, this restricted gas diffusion is also what allows plants to sense submergence through the gaseous plant hormone ethylene. So ethylene is normally produced in almost all plant cells, and because it's a volatile and there's no metabolic pathway to degrade ethylene, it will normally the homeostasis is basically through production and passive diffusion into the atmosphere, and this normally keeps internal ethylene levels quite low. Now, when the plant is submerged, this gas will still be produced and will be passively trapped and can function as a really rapid and potentially a reliable signal for plants to sense that they are submerged and potentially anticipate the impeding low oxygen conditions. Now, not surprisingly, ethylene has actually already been shown for decades to regulate all kinds of adaptive responses during flooding that help the plant to avoid hypoxia. Such as maybe the snorkel response, and this helps the plant to for instance, restore reaeration with the atmosphere. However, the role of ethylene in metabolic anticipation and acclimation has actually hardly been studied, but I think we believe that this could be very important because many crop species especially do not have these hypoxia adaptive or avoiding responses. So actually recently, or in the last decade, we have found that ethylene is very essential for hypoxia tolerance and anticipation. So the type of experiments that we conducted is basically like this. So what we do is basically treat plants with a very brief treatment of 4 hours with a low but sufficient dose of ethylene or an air control. And what we subsequently do is basically choke these plants with 100% nitrogen to mimic hypoxic conditions. And what you see is if plants go into hypoxia with an ethylene signal, they can survive much better. So this is looking at root tip survival of Arabidopsis seedlings, but we see the same for many Arabidopsis accessions, also at the shoot level, and also different other plant species. Now, basically the my entire career has so far focused on trying to understand how this works. So how does ethylene lead to this enhanced hypoxia tolerance? And I hope to hopefully enlighten you on this today. So the first thing we wanted to look at is what we know from literature is that there's a certain set of genes called core hypoxia genes which help plants to acclimate to hypoxia because they are expressed upon hypoxia. And for me, it was natural to look at these genes first. So what we did is, again, we treated these plants with a brief treatment of ethylene. And you see basically that many of these genes, so I'm showing you two here in shoots and in roots in Arabidopsis. And basically that ethylene alone has almost no effect on the transcription or the mRNA levels of these hypoxia genes. But when you then switch these plants to hypoxia after 4 hours, you see that there's this drastic augmented response, basically higher and faster expression of these hypoxia genes. And we think that this coordinated response could help the plants to survive hypoxia better after an ethylene treatment. But there's something interesting going on because it basically seems like there's some kind of transcriptional switch that's being sensitized by ethylene. So what is this transcriptional switch? And here's where the proteostasis angle comes in. So what we know is actually that these hypoxia genes are regulated by oxygen sensing transcription factors, the so-called group 7 ethylene response factors. And there's 5 in Arabidopsis, and what they have in common is that they have this N-terminal motif of methionine cysteine. And the cysteine was shown to be able to be oxidized in already 10 years ago in a publication, and this functions basically to an oxygen sensing pathway. And how does this work? So these proteins are constitutively being translated and being produced. And when there's oxygen and also nitric oxide in the environment, the endagron pathway, so the PRT6, this is the E3 ligase, PRT6 endagron pathway, targets these group 7 ERFs, basically oxidizing the cysteine, the exposed cysteine, and flagging them ultimately for ubiquitination, meaning that when oxygen, NO, or the enzymes of the endagron pathway are available, that these ERFs are constantly being produced and constantly being degraded. But what was discovered is that when either oxygen or NO, or when you genetically remove any part of the endagron pathway, that this can no longer occur or it is reduced, and these transcription factors basically start to stabilize and can activate downstream responses such as these core hypoxia genes, and in turn facilitate hypoxia survival. So we wonder, does ethylene affect the abundance of these transcription factors? And what we found is that actually using various methods is that ethylene is able, independently of transcription, to increase the proteins of these transcription factors. So we're looking at RUB 2.12 here, and we also looked at various other versions of the group 7 ERFs, and we see that they stabilize in response to ethylene. And this was kind of surprising because we give these ethylene treatments at ambient oxygen level. So there's ambient oxygen, and still when we give ethylene, these proteins seem to stabilize. So how is that possible? And what we know from a paper from Daniel Gibbs, so the lab where I'm currently working on with Dan, is that they showed not only do you need oxygen to destabilize these proteins, but nitric oxide is also crucial in this pathway. So I asked the question, does ethylene affect nitric oxide levels? And basically in literature, there were already some indications that there are strong crosstalk between ethylene and NO. So I conducted an experiment basically looking at NO levels, using a fluorophore dye that becomes photostable and emits more fluorescence basically as it reacts with nitric oxide. So it is called DAF-FM. And under normal conditions, you see this typical pattern in the root, quite some fluorescence. But when we pre-treated plants with ethylene, or when we treated plants with ethylene, and you look at the same, you look at the NO levels or the fluorescence in the roots, you see that this is strongly reduced after 4 hours. So this kind of suggests that ethylene may indeed be able to reduce NO levels, at least in the root tip. So what are the implications of this nitric oxide decline? So we asked ourselves, we see that ethylene stabilizes these transcription factors, and on the other hand, we see that ethylene reduces NO levels. So are those two linked, or is this just a correlation? And the way we tested that, so here we have again a protein fusion reporter line for RAP2.12, one of those oxygen sensing proteins. And if you give ethylene, they stabilize. Now, is this because of a reduction in NO? And the way we tested this is by the end of the treatment of ethylene, we give a pulse with nitric oxide at the same time. And what we see is that we can reverse the stabilization. So these proteins can again be degraded, suggesting that the increase in stability is because of a lack of NO, which ethylene basically facilitates. And in the other way around, we could also give an artificial NO scavenger called CPTIO, and we could stabilize these proteins independently of ethylene under normoxy conditions. And to verify that basically that these root tips are still viable, and as a nice positive control, we treated all of these treatments also after with hypoxia, as it was discovered basically, and we could show that the root tips are viable and they can still stabilize these proteins. And so this is RAP2.12 using a fusion protein fusion with GFP, but we also did similar things with RAP2.3 using a Western blot with HA tag. And we see some very similar responses, basically, as you can see here, compared to here. So for me, the latter part of my PhD was asking the question, how does ethylene then mediate NO levels? And what we know from literature is that there is a certain group of proteins called plant hemoglobins, or so-called phytoglobins. And these are very potent scavengers of nitric oxide in plants. And we found that ethylene, in fact, upregulates, in Arabidopsis at least, one of them, phytoglobin 1, already after 1 hour of treatment. And this is transcript levels, but we could see the same with protein levels. So we wondered, okay, that looks again like a really nice correlation. So is this upregulation of phytoglobin needed to reduce the NO levels in plants and basically induce this pathway, the signaling cascade? And for that, we identified a tDNA line, which has a tDNA in the promoter of phytoglobin 1, and that makes it insensitive to ethylene signaling. So phytoglobin is still there, but somehow it is no longer able to induce phytoglobin in response to ethylene. And when we then look at NO levels again, we see that ethylene no longer is able to reduce NO levels, basically showing that the upregulation of phytoglobin 1 is essential, or is needed to modulate these NO levels. And in the paper, you can also find, so due to restriction of time, I'm not going to show all the data today, but you can also find that this upregulation of phytoglobin is essential for the stabilization of the group 7 ERFs downstream, the augmented response of the hypoxia genes, and also the enhanced hypoxia tolerance. Because without this, in this mutant, ethylene is no longer able to do that. So with these results, we came to a proposed model to summarize basically a bit in the middle, which I think is rather detailed and elegant signaling cascade of how this could potentially work in nature. So under normal circumstances, we see that plants emit ethylene because it's constantly being produced, but However, upon submergence, you see that this ethylene can accumulate and leads to really rapid ethylene signaling within 1 hour. And actually, we see that within 1 hour, this leads to already upregulation of phytoglobin 1, and within 4 hours, we see a decline of the NO levels in the plant cells. And this is to such an extent that apparently it limits the degradation of this group 7 ERFs, and they start to stabilize. And we start to even see them accumulate in the nucleus. However, we don't really see a drastic upregulation of these target genes until we actually have a decline of the oxygen levels. So in this way, ethylene kind of sensitizes the hypoxia response, but only activates it when hypoxia is essentially there, kind of priming the plant, let's say, through this signaling cascade, basically coupling ethylene signaling to the hypoxia response. So this is, I think, a quite nice signaling cascade, but what can we actually do with this? So what does it mean basically in the bigger picture? So one of the other chapters that I did in my thesis is, can we explain why some plants are more flood tolerant compared to others? And is that explained by by the fact of whether ethylene can augment hypoxia response through this mechanism or not? So in Arabidopsis, we have this whole signaling cascade basically falling like dominoes in this direction, increasing tolerance in the end. So we looked at 2 plant species, so bittersweet and a more waterlogging variety of waterlogging tolerant variety of potato, and we could see that the same cascade could actually be induced and also leads to higher tolerance in bittersweet and also in potato. But in a more sensitive variety of the potato and also in tomato, we saw that in some cases ethylene was not able to induce phytoglobin and NO scavenging, and this also correlated with the absence of an augmented hypoxia response, and even in an absence being able to increase hypoxia tolerance in tomato. And we're currently doing something similar in barley, where we're looking at elite cultivars that may have lost this coupling of ethylene signaling to the oxygen sensing pathway. And I think in the future it would be worthwhile to see if we can restore this or reintroduce this to improve tolerance, basically. So there's one more question which we're still struggling with, and that is, while we know this upstream signaling cascade now, we don't actually really know what are the functional processes downstream that cause the plant to do better, or hypoxic cells to do better after an ethylene treatment. Which responses are doing that? Because I think we need to understand these really well to improve tolerance, basically. Now, in order to test that, we did several omics experiments. So we did transcriptomics and proteomics, and we tried to identify the underlying processes that are regulated by ethylene. So we did proteomics with Freddy Teodulu and Hongtao Zhang of Rothamsted. Today I'm not going to show that, but I'm going to show a little bit of the transcriptomics, which is mostly carried out by Ziguang Liu and Hans van Veen of Utrecht University. So we gave ethylene treatment followed by hypoxia again and reoxygenation, and we looked at the transcriptional response in time. And what we see is basically that you have these how do you call it, like the behavior of this gene expression follows these kind of patterns, and we could link this to different Go terms and processes, and also identify the underlying genes. And many of these things already confirmed of what we had found before, where ethylene basically enhances transcripts that are associated with flooding responses and ethylene, including the phytoglobins and hypoxia genes and group 7 ERFs. But we also found a very strong signature for ROS signaling and ROS scavenging, so the amelioration of reactive oxygen species and basically antioxidant activity like peroxidases. And on the other hand, we found a very quite a strong reduction in transcripts that are associated with development, so and especially root development and growth, and also with general translation. Maybe some of you root development people will recognize some of these interesting genes. So we're currently investigating this to much greater detail and trying to link these processes to tolerance and preparing. Hopefully I can show or we can publish a preprint in the near future. But what I want to focus on one thing is the antioxidants. So the antioxidant activity that appears to be regulated by ethylene in some aspect, and link it again to this upstream signaling cascade that I discussed before. So what we found is that that ethylene is able to reduce the formation of the excessive formation of reactive oxygen species during hypoxia and reoxygenations, and these are very damaging. So we wondered, is this why ethylene can protect plants from cell death, basically? And we tested this also in another way, is by treating with ethylene, or treating the seedlings again with ethylene compared to control, and then exposing the plants to high levels of hydrogen peroxide and see if they would survive better after an ethylene treatment, basically removing the hypoxia and linking it to the high levels of reactive oxygen species. And we find similar phenotypes. So here you have the wild types again, where ethylene improves tolerance, but similar to the hypoxia tolerance, we find that mutants impaired in phytoglobin or in the group 7 ethylene response factors, so the oxygen sensing pathway, have a reduced survival after this hydrogen peroxide treatment, basically showing that ethylene improves oxidative stress tolerance during hypoxia and reoxygenation, but partially through this upstream signaling cascade. Now, I want to summarize already a bit. So this suggests actually that there are many reasons for plant cells, why plant cells might die during hypoxia, and that there is actually a multitude of processes that need to be activated to ensure survival. And we find actually that ethylene alone already regulates a plethora of these responses. And we see that that hypoxia response and the antioxidant response are partially mediated through this upstream regulated mechanism, this oxygen sensing mechanism. And on the other hand, we find that ethylene may improve tolerance through a reduction in root growth and development and other metabolically expensive processes. So it seems like ethylene is like an early stirring signal to shift the balance from growth and regular growth towards adaptive responses, basically. I think that looking at all these different processes in the future can help us to understand better where we have to pinpoint the signaling cascades and where the specific processes we have to target in a case-by-case, in a case-by-case to improve tolerance, basically looking at a crop or whatever a tissue. So I just wanted to note that currently we're still linking the lack of the cessation of root growth and development in response to ethylene. We're still trying to link this to tolerance, and this is being carried out by Melissa in Utrecht. So I think this slide actually quite well summarizes the work that I've been doing in Utrecht, but I would like to take one more slide and one more minute to explain what I'm actually currently doing and what I'll be doing in the future. So, and this all has to do with the protein vernalization 2, VERN2. And now VERN2 is typically known as a subunit from the polycomb repressive complex 2, and this is required for the memory of winter through epigenetic regulation of by placing histone, by playing methylation mark on histone 3. And actually, VERN2 was shown by Daniel Gibbs by this lab to be so VERN2 is like the group 7 years. VERN2 is a target of the PRT6 endegron pathway, and then showed that VERN2 stabilizes through the endegron pathway or inhibition of the NF2 pathway, that it stabilizes in response to low temperatures, but also hypoxia, submergence, and the removal of NO. So very similar to what we see with ethylene. In addition, this has functional consequences because we see that stable VERN2 actually improves stress tolerance, specifically flooding and hypoxia tolerance. So that's, I think, very interesting. In addition, we show that VERN2 is actually slowing development. So stable VERN2 leads to a slower development, and removal of VERN2 genetically actually accelerates different aspects of development, such as primary root growth and hypocotyl length. And basically, if you sum all these things up, it looks like VERN2 is an epigenetic regulator of the processes that I just described that are that we see are regulated by ethylene during flooding and hypoxia stress. But because VERN2 is an epigenetic modulator, it has the capacity to tune this trade-off basically between stress responses and growth and development in the long term. And I'm currently trying to investigate how is VERN2 regulated during submergence, what are the target genes of VERN2, and does it have the capacity to mediate long-term flooding tolerance and potentially flooding stress memory in the current lab of Dan Gibbs. And with that, I would like to end. I would like to thank the plant ecophysiology group at Utrecht University, where I spent almost 7 years doing research, my PhD, master's, and a little bit of postdoc. Had a great time there, and yeah, if you ever have the opportunity to work there, I would certainly recommend it. I need to thank my mentors, Rensen and Rashmi, my direct colleagues, and the students that I had the privilege of supervising. In addition, I want to thank all the collaborators and my current lab, so Dan, Anne-Marie, and Rory, also Freddie for working with me on the proteomics and being, I think, a good friend by now, and the funding agencies and the companies that all made it possible. And I want to thank you for your attention and taking a little interest in my work. And if you have any questions, you can ask them now, but if you're watching this back later on YouTube or whatever, then please just contact me via email or Twitter or any of the social media means. So thank you.
Thank you very much, Sjon. Fantastic talk. Just checking the questions. Okay, the first question I'll ask is from Mirko Zafanini, and they'd like to inquire whether you know the relationship between ethylene and nitric oxide generating systems.
Sorry, could you repeat the question?
They would like to inquire whether you know the relationship between ethylene and nitric oxide generating systems, which are still elusive in plants, they add.
Yeah, so we wrote a bit of a part of a review of that in Plant Fish. So there are actually a lot of interactive and contradicting data out there. And I think that the contradiction comes mainly from looking at the timing and spatial dynamics of these things. So if you look at the literature there, if you give ethylene, the ethylene NO could go down or can go up. If you give NO, ethylene can go up or can go down. So, and it depends on the time scale and the plant species and the organ and the developmental stage that you look at. And there are already quite some molecular underlying mechanisms. So there is some evidence of ethylene modulating nitric reductase activity, which mediates NO production. There's evidence, of course, of phytoglobins here. And there's some evidence, I think, of NO directly affecting ethylene biosynthesis, both in a positive and a negative pathway. But directionally, it really, I think you have to test in your own system what's going on there.
Okay, thanks. I actually have a related question, and that was, do you know if you take Arabidopsis mutants that have altered nitric oxide levels, do they show any hypoxia tolerance phenotypes?
So our overexpression line of hemoglobin 1 has much lower NO levels than control plants or wild type plants, and they are consistently throughout literature been reported as much more flooding and hypoxia tolerant, like in many species. So that looks like it, but this links it to phytoglobin. So there are other mutants like biosynthesis mutants, and they seem to be, I don't really know for sure. I think there are not real clear phenotypes on those, but I think they have other issues as well. So there are many ways to produce or scavenge NO, and you are probably mediating or meddling with other processes as well.
Yeah, very true, I think. We have a question now from Kate Durley. She says, thank you for the fascinating talk. Nitric oxide also has beneficial effects on hypoxia survival. Yeah. I was wondering, is it known how ethylene-dependent nitric oxide depletion is balanced in cells in order to allow the beneficial effects of nitric oxide while also preventing
so I've been So I've been struggling with this myself, and I think it's a timing thing. So what we show is that if you have lower NO before going into hypoxia, it seems to be beneficial because these ERFs are stable. But it's also been shown that during hypoxia, you have a massive NO burst. So just at the onset of hypoxia, you have a massive NO burst, and this seems to be adaptive because it nitrosylates all kinds of proteins that are probably important for flooding tolerance. And if you limit this NO burst during hypoxia, this seems to be very negative for flooding and hypoxia tolerance. So again, it has to do with the timing and the experimental setup, I think, where we're looking at. Yeah, I think that would be my answer here.
Okay, thanks, Sjon. Maybe we can squeeze in one final question, and this is from Sangeeta Sapkota. They're asking, did you find any relationship between ethylene and reactive oxygen species as a signal, in parenthesis positive?
So I didn't look at that directly, but I know that ethylene regulates things like NADP oxidases, and that ROS signaling is definitely important for flooding tolerance, like NADP, like ARBO-HD, things like this are important for hypoxia acclimation. But we didn't look into this, but there's quite some literature out there looking at this, yeah.
Okay, thanks very much, Sjon, and thank you to Sophia as well. So that's 1 hour since the session started. So I think now I'll hand back to Katie Rogers from ASPB. Thank you very much.
Yeah, thank you very much, Michael, for doing a great job with moderation. As Sjon said, he's very active on Twitter, so I'd encourage you to ask your questions there too. Thank you all for joining. We'll be having 2 more talks in the Plantae Present series for this season, and then we'll be switching over to the plant biology meeting. So I'd encourage you to check that out if you're interested in more research talks. Browse the website and see if there's anything you're interested in joining on with that. Yes, thank you again for joining, and we'll see you at the next one. Bye, everyone.